Hello, good morning, students. Today we are studying about types of cleavage. विदलन के प्रकार के बारे में स्टडी करेंगे. टाइप्स ऑफ क्लीवेज में बनने वाले ब्लास्टोमियर्स के फेट के आधार पर दो प्रकार का होता है क्लीवेज, डिटर्मिनेट क्लीवेज एंड इनडिटर्मिनेट क्लीवेज. डिटर्मिनेट क्लीवेज, also called mosaic cleavage, is in most protostomian members. It results in the developmental fate of the cells being set early in the embryo development. Each blastomere produced by early embryonic cleavage does not have the capacity to develop into a complete embryo. निर्धारी प्रकार का विदलन कहलाता है ये, और इसमें जो बनने वाले ब्लास्टोमियर्स होते हैं, इनमें यदि इन सबको अलग कर दिया जाए या सेपरेट कर दिया जाए तो प्रत्येक ब्लास्टोमियर जो है वो नष्ट हो जाता है, वो आगे डेवलप नहीं हो पाता है, उसमें इतनी कैपेसिटी नहीं होती है कि वो पूरा कंप्लीट एम्ब्रियो बना सके. तो दिस टाइप ऑफ क्लीवेज इज डिटर्मिनेट क्लीवेज और निर्धारी विदलन. इनडिटर्मिनेट क्लीवेज, a cell can only be indeterminate, also called regulative, if it has a complete set of undisturbed animal/vegetal psycho-architectural features. It is characteristic of deuterostomes. This type of cleavage is salient feature of deuterostomes, indeterminate cleavage. When the original cell in a deuterostome embryo divides, the two resulting cells can be separated and each one can individually develop into a whole organism. If we separate out the blastomeres in this type of cleavage, each blastomere has the capacity to develop into a whole organism. It means each blastomere is able to form the whole organism whether they are separated out. Next, in this slide we can understand the difference between determinate and indeterminate cleavage. Indeterminate cleavage, we can see that if these cells are separated from each other, each has the potential to develop into an entire organism on its own. While in determinate cleavage, if these cells are separated from each other, they will die. Neither has the potential to develop into an organism on its own. In determinate cleavage, if these blastomeres are separated, then they will be destroyed. They will not develop into the whole organism. Now, on the basis of the extent of cleavage, extent of division, the cleavage is of two types: holoblastic cleavage and meroblastic cleavage. Holoblastic cleavage is further divided into following categories: bilateral, radial, spiral, and rotational, while the meroblastic cleavage is mainly of two types: discoidal meroblastic and superficial meroblastic. So in this slide we see that holoblastic and meroblastic cleavage. Holo means complete, whole. Mero means in fractions, in part, अंश भंजी. holoblastic in Hindi, पूर्ण भंजी विदलन. Holoblastic, the whole ovum is divided, while in meroblastic only at the animal pole where the yolk is less and where the yolk is in rich concentration, the embryo remains undivided. So this is the basic difference between holoblastic and meroblastic cleavage. In holoblastic cleavage, the zygote and blastomeres are completely divided during the cleavage, so the number of blastomeres doubles with each cleavage. In the absence of a large concentration of yolk, four major cleavage types can be observed in isolecithal cells: cells with a small even distribution of yolk, or in mesolecithal cells or microlecithal cells, moderate amount of yolk in a gradient, bilateral holoblastic, radial holoblastic, rotational holoblastic, and spiral holoblastic cleavage. These holoblastic cleavage planes pass all the way through isolecithal zygotes during the process of cytokinesis, Coeloblastula is the next stage of development for eggs that undergo these radial cleavaging. In holoblastic eggs, the first cleavage always occurs along the vegetal animal axis. Along the vegetal animal axis of the egg. The second cleavage is perpendicular to the first. From here, the spatial arrangement of blastomeres can follow various patterns due to different planes of cleavage in various organisms. The first subcategory of holoblastic cleavage is bilateral cleavage. Bi means two, di means two, and lateral means side. The first cleavage results in bisection of the zygote into left and right halves. It means due to the first cleavage, the zygote is divided into two halves, the left half and the right half. The following cleavage planes are centered on this axis and result in the two halves being mirror images of one another. And these two halves are the mirror images of one another. In bilateral holoblastic cleavage, the divisions of the blastomeres are complete and separate compared with bilateral meroblastic cleavage in which the blastomeres stay partially connected. Radial versus spiral cleavage. So in this slide we can see the difference between radial and spiral cleavage. In radial cleavage we see that zygote is divided into two blastomeres due to first cleavage which is along the animal vegetal pole, and this makes two blastomeres. Then these two blastomeres by further divisions makes four cells, and this is first quartet of the cells. Now these four blastomeres further divide and makes second quartet lies directly on the top of the first quartet. So now we have 8 cells. While in spiral cleavage we see the first division is along the animal vegetal axis. It is vertical, 2 cells are made. Then from these two blastomeres 4 cells are made. This is also the first quartet of cells. But here the difference begins. While in the radial cleavage, the first quartet of cells, it means there are 4 cells, divides horizontally and makes 4 blastomeres on the top of the 4 blastomeres at the bottom, equal size, while in this spiral cleavage, the second quartet lies in depressions between the first quartet. So top cells are micromeres and bottom cells are macromeres. The size is also different. The size of the top quartet is less than the size of the bottom quartet. So the top quartet cells or blastomeres are micromeres, while the bottom side quartet cells or blastomeres are called macromeres because they are greater in size. Radial cleavage. Radial cleavage is characteristic of the deuterostomes, which include some vertebrates and echinoderms in which the spindle axes are parallel or at right angles to the polar axis of the oocyte. Spiral cleavage. Spiral cleavage is conserved between many members of the Lophotrochozoans taxa referred to as Spiralia. Most spiralians undergo equal spiral cleavage, although some undergo unequal cleavage. This group includes annelids, molluscas, and Sipancula. So these are the examples of animals in which spiral cleavage is found: annelids, molluscas, and Sipancula. Spiral cleavage can vary between species, but generally the first 2 cell divisions result in 4 macromeres, also called blastomeres, each representing one quadrant of the embryo. These first 2 cleavages are not oriented in planes that occur at right angles parallel to the animal vegetal axis of the zygote. At the 4 cell stage, the A and C macromeres meet at the animal pole, creating the animal cross furrow, while the B and D macromeres meet at the vegetal pole, creating the vegetal cross furrow. With each successive cleavage cycle, the macromeres give rise to quartets of smaller micromeres at the animal pole. The divisions that produce these quartets occur at an oblique angle that is not a multiple of 90 degree to the animal-vegetal axis. Each quartet of micromeres is rotated relative to their parent macromere, and the chirality of this rotation differs between odd and even numbered quartets, meaning that there is alternating symmetry between the odd and even quartets. In other words, the orientation of divisions that produces each quartet alternates between being clockwise and counterclockwise. With respect to the animal pole, the alternating cleavage pattern that occurs as the quartets are generated produces quartets of micromeres that reside in the cleavage furrows of the four macromeres. When viewed from the animal pole, this arrangement of cells displays a spiral pattern. Now, rotational cleavage. Rotational cleavage involves a normal first division along the meridional axis, giving rise to two daughter cells. The way in which these cleavages differs is that one of the daughter cells divides meridionally, whilst the other divides equatorially. Mammals display rotational cleavage and an isolecical distribution of yolk, sparsely and evenly distributed. Because the cells have only a small amount of yolk, they require immediate implantation onto the uterine wall in order to receive nutrients. The nematode Caenorhabditis elegans, a popular developmental model organism, undergoes holoblastic rotational cell cleavage. Now in this slide we see the naming pattern or numbering system for spiral cleavage. E.B. Wilson developed a numbering scheme to follow individual cells through spiral cleavage. Macromeres and micromeres are grouped into quartets and numbered according to first quartet ancestor and number of cell divisions. Allows embryologists to follow cell phase and compare development in various taxa. Thanks for watching.